So, Stéphane, the floor is yours.
Thank you very much. So as you just said, my talk will focus today on sampling considerations in the context of Bayesian models in phylogeography. So as you all know, it's now fairly easy to access georeferenced genetic sequences. But if you simply just count genetic variants in different places across your habitat, then you're not going to be able to quantify dispersal. If you want to do so, you need to incorporate a temporal dimension, and phylogenetics allows you to just do that. And once you define your model in this context of phylogenetics, of evolution, then you're going to be able to estimate dispersal, and you're going to be able to do all sorts of interesting inferences, including dating past migration events, which is fairly relevant from a biological standpoint. So the idea of incorporating spatial information into models of evolution goes back to the 50s with the work of Wright and Maleko on the isolation by distance models. And in the first version, their model applied to the case where habitat was considered as a continuum. But later, Joe Felsenstein showed that this model had some sort of built-in inconsistencies, which led to the formation of larger and larger clumps of individuals, making that IBD model not very realistic from a biological point of view. So people then decided to discretize space in order to alleviate this clumping issue. And they came up with those migration matrix models, such as the stepping stone and the island model. And for those models, the population density is very strongly regulated because it doesn't vary at all within each dim. It doesn't vary stochastically or deterministically. And interestingly here, most inference techniques that rely on those models do not really deal with sampling patterns here. So it doesn't matter whether the dims I've sampled are next to one another or whether they are scattered across the habitat. It won't make any difference for the model, for the inference, sorry. The structured coalescent was proposed in the 90s. So here it's a backward in time model. So that's what really makes the sampling easier to deal with, all right? Here again, there's no random variation of population size, but you can incorporate deterministic variation of dim sizes. The structured coalescent is fairly demanding from a computational perspective, although there are some recent improvements here. But an issue I wanted to mention here again is about sampling, because the first problem you can notice is that sometimes the way you delineate dims is not fully driven by the biology of your organism. It could appear to be fairly artificial, right? That's the first issue. The second issue is that sometimes you have ghost dims that you haven't sampled, and we know that those dims may have a negative impact on the inference of migration parameters, for instance. So in 2016, with two colleagues from the University of Auckland in New Zealand, we've suggested to use a distinct type of coalescent, where here the habitat is considered as a continuum. And so that allowed us to somehow fix those issues with the sampling. but still the solution we proposed is fairly involved from a computational perspective, right? Now, if we move on to the next two models, the migration model proposed by Lemay and collaborators in 2009. So here we have a continuous time Markov chain to describe how genes are going to jump across different DIMs. So the setting is fairly similar to the structured coalescent, and what we just did here is that we flipped the direction of time around. So it's fairly fast to calculate probability densities under this model. The problem here is that the inference relies on a standard coalescent, and that's completely at odds with the fact that we are dealing here with a population that is structured in the first place, right? So there's no panmixia going on. Ghost DIMs and the contours of your DIMs of your subpopulations is also an issue. And in 2015, Nicolas de Mayo and collaborators have shown that migration parameter estimates may be strongly biased depending on how locations are sampled throughout your habitat. Now, moving on to the last model I wanted to talk about in this overview. We have this time a forward in time model, just like the previous one. And the big difference here is that the coordinates of the lineages are supposed to evolve according to a Brownian process running along the lineages of your tree. So it's here again a fairly easy to deal with from a computational perspective. So it's fairly easy to calculate probability densities. Here it's flexible because we have this Brownian process going on. However, there's no clear mechanistic underlying interpretation of that model. And it's not clear to me whether there's a convergence here to a Kingman coalescent in this particular model. In 2020, so just last year, that's in a collaboration with Nicolas de Mayo and his colleagues, we've shown that here again, just like for the migration model, we have biases problems when we look at migration parameter estimates and depending on how lineages are sampled in space. All right, so the two main points I wanted to convey here in this overview is that first of all, discretizing space may lead to some issues, but to me the most important one is that the current methods do not deal properly with patterns in the way lineages are sampled in space. All right. So the work we did here is, so we started from using the model I just told you about, the Brownian motion model, and we modified this model in order to incorporate two different sampling schemes, all right? And we're going to see that for one of those two schemes, we needed also to develop specific statistical inference techniques to infer the parameters of this version of the model. All right, so here I'm going to go through the two different sampling schemes carefully. So that's probably the most important part of the talk here. If you understand how those two sampling schemes differ, I'm going to be very happy about that. All right, so first of all, the detection scheme. Here you assume that the places of sampling, so the places where you collected your samples, those places are purely selected based on the outcome of the migration process running along the tree, right? So you assume here that you have a Brownian process going on running along the tree, And at some point in time, you observe the outcome of this evolutionary process. So you have this first random variable represented here by those little black disks. So those are the outcome of the evolutionary process. And you have then sampling going on. And sampling is considered here also as a random process. And it's depicted here with those purple circles, right? And here the sampling exactly matches the outcome of the evolutionary process. So that's the first sampling scheme. The second scheme is the survey scheme. And here you assume that evolution has been going on for quite a while. And you have lineages scattered all over your habitat. But for some reason, some places for sampling lineages are easier to access compared to other, all right? And here what's important is that the places you go to for sampling do not depend on directly on the outcome of the evolutionary process, right? So here you could, it could be the case that this upper corner here is easier to sample compared to the other places in your habitat, so that you will have an excess of samples collected here, all right? So those are my two different schemes, and I'm going to try to set up a Bayesian model based on those two different approaches. All right, so if we start with the detection scheme, so we obviously have a posterior. And so I'm going to go through quickly the different random variables involved here. So first of all, we condition on the data. So the data here is made of a set of homologous sequences aligned. And we also have observed sampling locations, right? The two parameters we are mainly interested in are the dispersal parameter, dispersal rate, and the effective population size, because as you're going to see, we're using a coalescent here. So we have this effective population size parameter along with the coordinates of your lineages along the tree. So that's a vector of coordinates where we observe the coordinates at the internal nodes of the tree and at the tips as well. And we have a time tree, which is here tau, and that's a phylogeny with branch lengths expressed in terms of calendar units. So we break down this posterior as usual by expressing it in terms of a product of the Felsenstein's likelihood, so the probability of the sequence alignment given the tree. We have the coalescent density around here. We have the prior density on the dispersal and the effective population size parameter. And what we are really interested in here is this part of the model where, first of all, we have here this density F, which corresponds to the forward in time density of the locations running along the tree. So that's the Brownian motion part of the model. And here, that's the sampling part of the model. So this is where we model sampling. So here, you remember what we said about the detection scheme. We condition the sampling based on the outcome L of the evolutionary process. So for this reason, we are using a delta Dirac function here. And once again, the outcome of the migration process, which is L, it fully determines E, the sampling locations. Now, for the survey scheme, we flip the conditioning around, right? So here we have this part of the model that describes the distribution of sampling location. And here the sampling is not conditioned on the outcome of the evolutionary process. So here would be a good place, for instance, to incorporate knowledge about the various intensities with which we've sampled different places in our habitat. And the second part here of the model has to do again with the outcome of the evolutionary process, but this time we need to be very careful here because we condition here on the fact that we've sampled organisms at particular places. And that's this conditioning that is going to get us into trouble, right? So that's what I'm going to focus on. From now, I'm going to focus on the product of this density. So the location given the tree and the sampling locations multiplied by the coalescent prior. So if we look at the way we need to define this density, because we're conditioning on E, on the sampling locations, we have to introduce that term, which is a normalizing term, that is a function of the dispersal parameter and the effective population size. And here, basically, what this normalizing term is somehow we need to sum over all possible genealogies and integrate over all possible node ages and internal node locations, so that what we observe at the tips is compatible with what we've sampled. And the point here is that the value of Z here is, we can't calculate it directly. We don't have a closed form formula for that, all right? And the problem we are facing here, therefore, belongs to the class of doubly intractable problems. So we have an intractable term here that is involved in the definition of the likelihood of the model. And of course, we also have another intractable constant that is involved in the Bayesian formula, right? So that's the denominator in the Bayesian formula. So that's a doubly intractable problem. And it turns out that I've worked on this sort of problem, but in a different context, in the context of molecular dating. And therefore, I was able to somehow transpose what I've learned on those sort of problem into the setting that we are interested in here today. All right, so in 2010, Murray and colleagues described the exchange algorithm that was designed to specifically tackle those doubly intractable problem. So here, it's probably best to rely on the graphical model to explain how this algorithm works. And the graphical model I provide you with here gives you a way to generate observations according to the model. It's not the best way, the most efficient way to generate observations, but it's a valid way from a statistical point of view. So what you would do if you were to use this graphical model would be first to select a set of pairs of values for the dispersal and for the effective population sizes. So you have here n pairs of values for those two parameters. And for each of those pairs of parameters, what you would do is you would generate a pseudo phylogeny and a pseudo set of locations along this pseudo phylogeny. So here, the way you generate this pseudo phylogeny and location is, well, you have set your effective population size. So you can use that to generate a tree according to Kingman's coalescence, and conditioned on your tree, conditioned on the dispersal parameter and conditioned on the sampling locations, then you're able to generate locations at the internal nodes of your pseudo tree and at the tips as well, of course. The third step consists in selecting a value for an index random variable, right? So I here is a value chosen from a discrete random variable distribution, and this distribution actually matches that of the prior distribution for the model parameters. And the last step, once you've done that, is to copy the corresponding pseudo tree and pseudo location to obtain the actual tau and L, the actual tree and locations. All right, so if you were to assign probabilities to each of these operations that you've conducted here to generate an observation, then you would obtain the joint posterior density that you're interested in, all right? So what this means is that if you're able to build a Markov chain which states correspond to different instances of this graphical model, then you're going to be able to sample from the posterior distribution of interest. All right, so the core of the exchange algorithm really is depicted on this figure here. So you have two instances of your graphical model, and the only difference you see between those two instances that in order to go from M to M prime, here you've just exchanged the values of the pseudo tree and the pseudo locations here. You've exchanged the pink part here with the blue part, and exchanging that gives you M prime, all right? So now if you consider the ratio of the corresponding probabilities, so the probabilities we've just seen before, so what you have in the numerator here corresponds to what you have for the model M. And of course, this is the posterior probability, so probability density. So as we've seen before, that involves the values of those problematic normalization terms, right? But if you look at the ratio between the probabilities, so the probability you get for M and that you get for M prime, then you see that those Z terms cancel out, right? So that really is the main insight behind Murray's approach here with the exchange algorithm. So it's through the introduction of this pseudo random variable X, so here the pseudo tree and pseudo location, that you manage to make those problematic terms vanish. Now, the last issue we need to deal with here has to do with those pseudo random variables, because the problem here is that we don't know how to obtain direct random draws from the corresponding distributions for those pseudo tree and locations. And because there's no direct way of sampling from the corresponding distributions, what we had to do is implement a Markov chain, a standard Metropolis-Hastings algorithm to generate valid random draws for those pseudo random variables. All right, so for the pseudo trees and pseudo locations, we obtain valid random draws by applying a standard Metropolis-Hastings type of algorithm. And calculating the acceptance ratio for this part of the algorithm doesn't involve calculation of the problematic Z terms. All right, so this slide here gives you the algorithm that is implemented currently for updating the value of the dispersal parameter. So this is just here one step of the MCMC that is used to get samples, correlated samples for sigma. So here you can recognize here the standard approach where we first sample the value from a proposal distribution Q, and here you have an acceptance probability that relies on a Metropolis ratio and a Hastings ratio. What differs here with the standard Metropolis-Hastings step is that you need to incorporate this MCMC within this Metropolis-Hastings step. So here, this MCMC is for updating the value of the pseudo tree and pseudo locations along that tree. All right, now I'm gonna move on to the results part. So this is the results of simulation work we did recently with Nicolas de Mayo. And what Nicolas did here is that he generated lineages location along a tree using a Brownian process forward in time. And we obtain here a population of lineages that are scattered along a rectangle. And what we do next is along the y-axis, we're going to sample a few lineages so 100 lineages, I think. We sample 100 lineages along the y-axis without any bias. So here sampling is done uniformly along the y-axis, but the sampling is biased along the x-axis, right? So here we've sampled lineages preferentially around this position 5 here.
Stéphane, sorry to interrupt, but your time is kind of up. I mean, so if you can just go a bit fast over this section, that would be great.
Okay.
Thank you very much.
So I'm just going to finish here by saying that the estimation of the location for the most recent common ancestor is still biased under the survey scheme, all right? But when you look at the diffusion rate estimates, what you observe is that you don't have any bias or virtually no bias anymore with the survey scheme, right? We've analyzed some real data sets as well, but I will tell you about this maybe at another time. So thank you very much for listening to me. I just wanted to insist once again on the importance of properly sampling and modeling sampling in the inference techniques in phylogeography. And I wanted to thank my collaborator on this project, Nicolas de Maillot, and the Agence Nationale de la Recherche that funded the Genospace project. Thank you.
Okay, so thank you very much, Stéphane, for this very interesting talk. So now we welcome questions. Please feel free to ask your questions in the chat. I think I will ask a question to start things. So actually, you know, I made you accelerate over that part, but you were showing these results where you compare the two schemes, the detection scheme and the survey scheme. Could you comment on, you know, like in the case of your simulated data, which one is more appropriate in the end? I mean, I guess you went fast, so I missed it.
Yeah. So here, the survey scheme seemed to be more appropriate because, as I said, we have a population of lineages that are uniformly scattered in your rectangle, in your habitat, and you decide to only collect lineages that are located in a particular place on that rectangle. And the way you do that is not driven by evolution. It's purely driven by the fact that it's just easier to get samples from that area.
I see. Okay. Yeah, thanks a lot. So, I mean, if we have questions, I would say feel free to speak up now. We don't see questions in the chat, so I'm happy to take.
I have a question.
Yes.
So I suppose, so here you assume that, I mean, things move like a random walk, but just at the end you had a real case, but a real case, I imagine that sometimes you may have some long range transport, and how does it play, how does it going to change the results?
Yeah, I suspect that it probably has an impact. What we would like to do in the next steps in this project is to combine different sampling schemes in a single analysis. We would like to say, okay, so those, so if you look at a pandemic, for instance, the first sequences you get are usually well described by a detection scheme, right? But once you get to a pandemic stage where you have observations all over the place, then it's best to use a survey scheme. So here, if you want to analyze the whole set of sequences, you're going to need to mix different sampling schemes. And if you know that some lineages move in a different way compared to other lineages, I suppose that you could probably incorporate this information in your prior, but there are probably other ways to proceed as well here.
Thanks. If there is any other questions, again, please feel free to speak up. No? I mean, I was also wondering actually the idea of discretizing space. You pointed out that it's problematic. So are there cases where it's okay, and is it easy to identify them a priori or not?
Well, if you work in species in Hawaii, for instance,
okay, right.
then it's fine. You know, I wouldn't mind using discrete dims here. But we've seen many examples where basically the different dims corresponding to different regions that were not defined based on biology. It was just defined based on history, basically.
Right, I see. Okay, cool. Well, thank you very much, Stéphane, for this great talk. And now we are going to move on to our next speaker.